Can everyone see my screen here?
Yes.
Yes.
Okay, so let's get started. Hi everyone. Today Yan and I are presenting for the Journal Club. We are going to introduce an article that includes two phase 3 trials about paclitaxel for the treatment of geographic atrophy secondary to age-related macular degeneration. And this topic is directed by Dr. Jebs. Before we dive into the two trials, I would like to provide some background information about geographic atrophy and age-related macular degeneration. So, geographic atrophy is an advanced form of age-related macular degeneration, AMD, resulting in severe irreversible vision loss in people aged 60 years or older worldwide. Geographic atrophy results in a high burden of illness, with quality of life severely reduced for patients and their caregivers. Consequences of visual impairments include increasing fall risk, difficulty in reading, driving, and recognizing faces, and ultimately loss of independence. In February 2023, paclitaxel was approved by the US FDA as the first treatment for the geographic atrophy secondary to AMD. Generally, AMD is a disease that affects a person's central vision, and it also affects the ability to see fine details. In AMD, a part of the retina called the macula is damaged. The severity of the disease varies. In its early stage, AMD may have no symptoms of signs, so many people may not suspect they have it. According to the Vision and Eye Health Surveillance System, in 2019, an estimated of 20 million Americans aged 40 and older were living with AMD. Of these, 1.5 million were living with vision-threatening AMD. The risk factors of having AMD includes age 50 years and older, smoking, high blood pressure, and high saturated fat diet. The two primary types of age-related macular degeneration have different causes. The dry form is the most common one. The real cause is unknown, although both genetic and environmental factors are thought to play a role. Dry form happens as the light-sensitive cells in the macula slowly break down, generally one eye at a time. The loss of vision in this condition is usually slower and gradual. The wet form is less common. However, it usually leads to more severe and faster vision loss in patients than dry AMD. Wet AMD happens when abnormal blood vessels started to grow beneath the retina. They leak fluid and blood and can create a large blind spot in the center of visual field. Geographic atrophy, GA, is a dry and advanced form of AMD. It is characterized by atrophied lesions that first started in the outer retina and progressively expand to cover the macula and the fovea, leading to irreversible loss of vision over time. About 1 million reported cases of GA in the United States, and approximately 106,000 cases occurs per year. GA is not diagnosed by laboratory or genetic testing. Diagnosis of GA is via examination and imaging. The best diagnostic technique for GA is fundus examination followed by FAF. Although vision function tests are not used to make diagnosis, they tell us the extent of visual impairment. Visual function can be measured using the following methods. The best corrected visual acuity is a standard vision assessment that uses Snellen chart or early treatment diabetic retinopathy study chart to measure visual acuity. It is measured by how many letters a patient can read at a distance. Reading speed provides information about how much the visual field is sufficiently preserved to recognize entire words or sentences as opposed to individual letters. With progression of GA, reading speed will decline. Microperimetry is an alternative to reading chart and may not use to measure retinal and may be used to measure retinal sensitivity in specific parts of a retina to identify functional and non-functional areas. Clinical features of GA are detected by using ophthalmoscopy and imaging. Common techniques that enable visualization of the fundus and surrounding vasculature include the following. The FAF detects retinal pigment epithelium loss and it's the primary imaging tool for tracking GA lesion size and growth in the clinical trials. OCT can also be used to identify GA lesions. However, OCT is better in managing wet AMD. The pathogenesis and mechanism behind treatment of GA are related. Overreaction of the complement system is implicated in the pathogenesis of GA. Genome-wide association studies have found strong association between genetic polymorphism in the complement pathway and AMD risk. Although the association between the complement system and GA expansion is being evaluated, dysregulation of complement proteins lead to retinal cell death through a sequence of chronic inflammation, opsonization of cells, and implication of the alternative complement pathway. C3 is the most abundant complement protein across complement activation pathway, and pegcetacoplan target at C3 and C3b, leading to its effect of improving cell survival and reducing vascular loss. The phase 2 trial shows that pegcetacoplan monthly and every other month significantly reduced GA lesion growth compared to the sham injection. So in the OAKS and DERBY phase 3 trials, the objective is to assess the efficacy and safety of intravitreal pegcetacoplan compared with sham for the treatment of GA secondary to AMD. OAKS and DERBY were similar to 24 months multi-center randomized double-masked sham control phase 3 trials. They were designed as confirmatory trials for regulatory purpose. In both studies, eligible patients were aged 60 years or older. Patients were included if their eye under treatment assessment had BCVA of 24 letters or better on ETDRS chart, or 20 and better on Snellen chart. The study eye needs to be diagnosed as GA secondary to AMD, confirmed by the reading center. The total GA area should be 2.5 to 17.5 square millimeter. If the lesion is multifocal, at least one lesion should be 1.25 square millimeter or larger on FAF imaging. For patients in whom both eyes met the study criteria, the worst seeing eye was the study eye, and the other eye would be the fellow eye. In OAKS, patients were also required to be able to perform the microperimetry assessment, which was not required in DERBY. Patients were excluded if the study eye had a geographic atrophy secondary to a condition other than AMD, and any history or presence of active choroidal neovascularization in the study eye will also be excluded. In both studies, eligible patients were randomized assigned to receive pegcetacoplan monthly or every other month, sham injection monthly or sham injection every other month for 24 months. The randomization methods was a stratified permuted block randomization. The stratification criteria were GA lesion area at screening, history or presence of active CNV in the fellow eye, and block size of 6. Both studies were double-masked. The study site staff, patients, reading center personnel, the assigned evaluating physicians, and the founders were masked to treatment assignment. However, the treating physician and any supported staff involving in performing the injection were not masked to the treatment. Individuals who were unmasked were not allowed to discuss treatment and patient outcome with study staff who were masked, including the evaluating physician. At 12 months, only the sponsor personnel responsible for analyzing, interpreting, and reporting data were unmasked to treatment assignment. The success of masking was not formally assessed. In both studies, a screening period of up to 28 days was followed by 24 months of treatment. For the first 12 months, all patients returned every month to the clinical site. From month 12 onwards, patients receiving pegcetacoplan monthly or sham monthly continue attending the clinical site on a monthly basis, while patients in the every other month groups attend the clinical sites every other month. The procedure of sham injection was the same as that used for paclitaxel, except for no actual injection occurred. Instead, the physician only touched the study eye with the blunt end of the syringe. FAF was done every 2 months, and images were evaluated by a minimum of 2 certified readers with independent manual measurement of features of the GA lesions. The reading center prospectively assessed fluorescence angiography for all patients at baseline, month 12 and month 24. BCVA was measured at every study visit, and other functional outcomes were measured every 6 months. An external independent data monitoring committee will review the data across the conduct of the study on ongoing basis. The primary endpoint was the change from baseline to month 12 in the total area of geographic atrophy lesions in the study eye based on FAF imaging. The key secondary endpoints were change in maximum reading speed of the study eye, change in mean functional study independent index score for both eyes, and change from baseline in normal luminance BCVA score. In OAK study, change from baseline in the mean threshold sensitivity of all points in the study eye by mesopic microperimetry was reported. Other secondary and subgroup analyses were changed from baseline to month 24 in the NEIVFQ-25 score, total area of GA lesions in the study eye at month 24, and change in lesion size stratified by subfoveal and non-subfoveal location based on FAF imaging from baseline to month 24. All of these endpoints were assessed using data from the combined studies. For OAKS, change in the number of scotomatous points in the junctional zone of atrophy from baseline to month 24 was reported. The main safety endpoints were the incidence and severity of ocular and systemic treatment emergent adverse events as months 12 and 24.
Thank you, Emily. Then I will introduce the statistical analysis and result part of the trial. In the study, patients in the sham monthly and sham every other month groups were put into a single control group for all analyses. Therefore, in the statistical analysis, we have 3 treatment groups: pegcetacoplan monthly, pegcetacoplan every other month, and sham group. Regarding power calculations, they assumed an overall standard deviation in change from baseline lesion area of 1.25 square millimeter per year, which based on natural history, and 1.5 square millimeter per year, which based on previous field study. As a result, a sample of 600 patients will provide over more than 99% power to detect a difference of 0.66 square millimeter in the change from baseline to month 12 in GA lesion area, and for a difference of 0.43 square millimeter. The power is about 92% and 81% respectively. And the power calculation was based on two-sided t-test at a significant level of 0.0495. Next slide. For the efficacy analysis, the efficacy analysis were done using the modified intention to treat analysis, where population was all patients who received 1 or more injections of pegcetacoplan or sham and had a baseline and at least 1 post-baseline value of the lesion area. They used the mixed effect models to measure the least square mean change from baseline to month 12 and month 24 with no imputation for missing data. Also, they combined OAKS and DERBY data to conduct the same analysis in the modified ITT population and in the subgroups with and without subfoveal GA involvement at baseline, which is a key predictor of GA lesion growth. About post-hoc analysis, they conducted piecewise rate of change to compare the slope of lesion growth with pegcetacoplan versus sham and linear trend testing was conducted to assess whether the treatment effect of pegcetacoplan increased over time within each of the 3-month period compared to sham. Additionally, key secondary endpoints and other secondary and subgroup analyses were performed by using the combined data. Two-sided significant level is 0.496. Next slide. Regarding the missing data, pre-planned sensitivity control-based multiple imputation analysis, post-hoc control-based multiple imputation sensitivity analysis were done to explore the impact of issues like study discontinuation, missing data, etc. Next slide. And for safety analysis, all patients who were randomly assigned and received 1 or more injections were included in the safety population, and safety data were recorded at month 12 and month 24. After screening, we see there are 637 patients in OAKS and 621 patients in DERBY were randomly assigned to 4 treatment groups. At month 12, about 89% patients in OAKS and 89% patients in DERBY completed, and at month 24, 74% in OAKS and 76% DERBY completed. So at the end, the modified ITT populations comprised 614 in OAKS and 597 in DERBY. The mean study drug compliance was higher than 89% at month 12 and higher than 87 at month 24. From Table 1, we could see that the demographic and baseline characteristics of the modified ITT population were generally balanced across the 3 treatment groups, no matter in OAKS or in DERBY. However, we could notice that in OAKS, pegcetacoplan groups had a higher proportion of patients who experienced the JA without subfoveal involvement compared to sham group. In contrast, in DERBY, the 3 treatment groups had a similar proportion of patients without subfoveal involvement. Given the fact that JA lesions with subfoveal involvement tend to progress more quickly compared to those without subfoveal involvement, it might partially explain why the 2 trials had different results, which OAKS met the primary endpoint, but DERBY did not meet the primary endpoint. Then I'll introduce the result of the primary endpoints. In OAKS, the primary endpoint at month 12 was met, with pegcetacoplan monthly showing a significant 21% reduction in total area of JA lesion growth compared to sham, and pegcetacoplan every other month showing a significant 16% decrease in JA lesion growth. In contrast, in the 12-month primary endpoint was not met in DERBY. And at month 24, in OAKS, the pegcetacoplan monthly and every other month slowed the growth of JA lesion by 20% and 22% and 18%. In DERBY, the drug monthly and every other month slowed the growth of JA lesion by 19% and 16%. The primary endpoint results were robust to missing data. Here we show the plots for primary endpoints. Plot A and B were from mixed effect models, and plot C and D were from piecewise linear slope models. We could see that at month 12, the OAKS trial suggested statistically significant treatment effect compared to sham, but the DERBY trial did not show a significant treatment effect. However, at month 24, both OAKS and DERBY trial suggested a statistically significant drug effect in slowing the rate of JA lesion growth compared to sham. Next slide.
Do you want to comment on the apparent dose differences in those curves?
I mean, the difference between for the OAKS, we see the at month.
So they didn't say they were going to compare the 2 different doses, but if you look at those curves, every other month does not look, well, it looks about the same as monthly, right? I mean, they look pretty close. So it's just, okay, sorry.
let's go ahead. Here we show the, here we use the combined data. The results show that the growth difference in J legends at month 12 was reduced 16% with pegcetacoplan monthly and reduced 14% with pegcetacoplan monthly every other month. At month 24, the difference is reduced 21% and reduced 17% respectively. And post hoc piecewise linear slope analysis showed increasing efficacy of pegcetacoplan over time. At months 18 to 24, the drug showed the greatest treatment effect with 30% difference in pegcetacoplan monthly and 24% difference in pegcetacoplan every other month. The next slide. Since foveal involvement is a key predictor of J lesion growth, with subfoveal involvement tending to progress more quickly than non-subfoveal, the study also conducted subgroup analysis by lesion location. Here we see non-subfoveal versus subfoveal using the combined data. For patients with non-subfoveal J lesions, the difference in lesion growth at month 12 were 26% in pegcetacoplan monthly and 23% in every other month. For patients with subfoveal lesions, the difference in lesion growth at month 12 were 11% in pegcetacoplan monthly and 8% in every other month, which were obviously less than the patients in non-subfoveal group. Other analyses showed that at month 24, no significant difference in other secondary outcomes, including visual function endpoints, a best corrected vision acuity, functional reading independence index score, monocular maximum reading speed, mean threshold sensitivity, and questionnaire 25 distance activity subscale scores. For safety analysis, the number of serious ocular treatment emergent adverse events in the study eyes was low in both OAKS and DERBY. For intraocular inflammation, 4 cases were linked to drug impurity. No event of intraocular inflammation led to permanent severe vision loss. Notably, the new onset exudative AMD was an issue related to pegcetacoplan. At 12 months, the new onset exudative AMD were reported in 11, 10, and 3 patients in OAKS and reported in 13, 7, 7 in DERBY. Compared to shame, patients with treatment of pegcetacoplan showed relatively higher likelihood to develop exudative AMD. Patients who developed exudative AMD continued study treatment, and most of them received anti-VEGF therapy. And when pegcetacoplan was co-administered with anti-VEGF treatment, no unexpected outcomes were observed, and no case of exudative AMD was reported as a serious adverse event. Here we summarize the key findings of the study. First, the study suggested pegcetacoplan slowed J lesion growth in the combined data compared to sham, with increasing effect over time. However, in the separate trial, OAKS met the primary endpoint, showing a statistically significant growth difference in J lesions at month 12, but DERBY did not meet the primary endpoint. The average magnitude of retinal preservation observed with pegcetacoplan treatment might be biologically meaningful. Although the study showed statistically significant difference in the change from baseline to month 12 in the total area of J lesions, there are no significant difference in other vision function measures. And the safety profile of pegcetacoplan was generally similar with that observed in other studies, but we noticed a higher incidence of exudative AMD in patients with pegcetacoplan treatment, which is a big deal for patients. There are some strengths and limitations of the study. The study included 2 separate multicenter randomized double-masked and sham-controlled trials, which separate and combined data used to conduct multiple analyses. For primary endpoint analysis, they use the modified ITT analysis, which provide a strong methodological foundation to reduce bias and improve the reliability of findings. And other subgroup and sensitivity analysis were also performed to provide insight further. Second, the large and diverse sample size, which over 1,000 patients involved across more than 100 clinical sites worldwide, strengthened the findings. Also, pegcetacoplan became the first FDA-approved treatment for GE, highlighting the trial's clinical significance and contribution to advance the treatment options. For limitations, first, the study included the high proportion of patients with subfoveal GA, which might have made the assessment of the effect on visual function more challenging. Second, the OCT angiographic imaging was not available at all sites, preventing a systematic evaluation of the presence of non-exudative neovascular lesion at baseline. Third, some missing data due to studies continuations and COVID-19 could be a potential source of bias. Then, next slide. Then we propose some discussions about the trial. For discussion 1, why did DERBY fail to meet significant level at month 12, but OAKS did, and how to evaluate the evidence from the study, which the combined data showed a statistically significant difference with OAKS meeting the endpoint, but DERBY not. Someone have some idea about this question?
Well, so, the results of these two trials led to the approval of the drug. It's the first drug, so there was no treatment for geographic atrophy prior to that. Anti-VEGF has been effective in the treatment of neovascular AMD, and it's widely used, and there's tons of trials on various anti-VEGF agents and regimens. So there was certainly a sentiment that this drug should be approved because there's nothing else. There are no options. But, I'd love to hear what people think about at the end of this conversation, what they think about whether the drug should have been approved. The law is that it's supposed to show efficacy and safety in two well-designed controlled studies. And here it showed it in one and not in the other. Now, in the other, it missed the primary endpoint. It sort of looked better at 24 months, but the primary endpoint was at 12. And then you have only one of the two showing the primary endpoint, hitting the primary endpoint, and you see no functional difference. So yeah, you've slowed the growth of the GA lesions, but there's no functional difference. And then you add that there's a higher risk of neovascular AMD with sudden visual loss, which is, on the order of about 5% per year. And it became somewhat controversial. Should it have been approved? And so, I don't know, maybe open it to either some of your colleagues or one of the faculty or some of the faculty. I know Anne, I'm sure will have some comments on this about how do you handle a situation like this? I mean, these are big trials. They're well designed. I mean, these guys went out of their way to deal with masking issues. And so, what do you think? Anne, any comments? Or did Anne have to run off? Looks like she did. Okay. Shariza, any comments on, oh, Dave had his hand up. Okay, Dave, go ahead.
Well, I'd certainly be happy to wait until students have had a chance to react if they want. So certainly the parallels are not perfect, but this reminds me of the situation with regard to the two Alzheimer's drugs that were approved. Those drugs had considerable safety concerns that I think are not quite applicable here, but the same thing, reasonable efficacy on some marker and little evidence that it helped the things that people care about most, which I think is an important question that I think you're also posing here, Doug.
Yeah. And, you know, this is more than a marker. I mean, this is actual destruction of retinal tissue. So it's a biologic event, but at the end of the day, does the benefit of the drug justify its approval and its use? And this is, again, it's a biologic that's injected monthly, and which means it's an expensive proposition that made no difference in visual acuity, reading speed, microperimetry, or anything else that indicates a physiologic benefit.
Yeah, I think fair to say my choice of the term marker was probably not great. Removing protein accumulated in the brain and removing and changing this outcome, both of which are not the things that I think patients care about. In the area of the Alzheimer's drugs, at least one of them has now been removed from the market. So even though the FDA approved it, the market spoke. And
Yep.
I think the other one is not doing well in the market, and I won't be surprised if it lingers for a while and then gets removed. And I wonder here, is there any evidence that people are now using this?
Well, a second drug was approved.
Okay.
I think what you hear from a lot of people is they might use it in somebody who had a fovea threatening lesion, because if you slow down the rate of progression towards the fovea, somebody who's got good vision, you might preserve the time period during which they maintain good vision. And one of the things is that in OAKS, there's a high proportion of patients with subfoveal lesions, and therefore, you might have difficulty showing improvement in vision in those people. So, I mean, I think that's one of the reasons why they pointed it out in the manuscripts, was to try to hedge their bets about that. Shariza.
Yeah, I'm gonna put on my policy hat just for a minute. I know we're talking trials here, but in terms of the general trajectory of FDA approvals, I've noticed, and I don't know if others have as well, a general trend towards approving primarily more so on safety than efficacy per se. And I think you've both kind of touched on that as well. But playing devil's advocate, I wonder if the part of the logic behind that is opening the doors for further investigation on additional indications for these projects, perhaps being done by academics, perhaps being done by others. I'm not sure if that's really one very small bright light in light of something like this.
For sure. Like, there's also increasing concern about the fact that the FDA derives more and more of its own operating budget from the
PDUFA.
companies it seeks to regulate.
Yeah, PDUFA, GDUFA, MEDUFA, go down the list of legislation.
Yeah.
Yeah. I would say, like Alzheimer's, I think the absence of any treatment at all probably makes them more sympathetic to a drug that looks like it's likely to work.
Right. I completely, I would agree with and share that concern. If you have no treatment, is it worth taking more of a chance on a potential treatment? But we don't have a really well-formulated policy about that. This seems rather hit or miss to me.
Yeah.
I don't know how controversial this approval was. In the Alzheimer's community, the other approval was very controversial.
I think the approval was less controversial. I think how much people are going to use it is, as you said, the market will tell.
Yeah.
And, I mean, can we go back to the data on the...
I don't know if you all see, Ian also had a comment / question.
Yes.
Yeah. I mean, if you look here, oops, no, that's fine. If you look here, nope, go up one. Yeah, right here. I mean, they miss, right? It misses at 1 year in Derby. It makes it at 2 years, right? And I'm sure that they argued that, Derby had a higher proportion of extrafoveal lesions and those grow more slowly. And therefore we goofed on the design. We should have said 2 years or whatever. And, it works at 2 years. And when you look at the graphs, the uncombined graphs, the combined graphs, I think are very suspect because, it could be all one study driving it and the other. But if you look at those graphs, it sort of looks like they're having benefit. It just, you didn't pick it up at 1 year to pick it up at 2. So, there is some sentiment here that, the primary outcome didn't missed in Derby, but boy, you still look at the data and it looks like it's working. It works. It's just not a great drug, but it seems to work. I don't know what people think about that sort of interpretation of it.
But then it begs the question, does this set a precedence that will then lead to a downward spiral in terms of what we consider efficacious?
I couldn't agree more. I mean, when these data came out and it was approved, I was scratching my head. I said, why do we do this? So, and I'd be interested from our students, how many think that that if you were sitting on the FDA advisory committee, how many think you would have approved the drug based on these data? Maybe we can have a show of hands and from the students and just let us see how many think they would have approved it. If you think you would have approved it, put your hand up. Now I see a couple. 3. Yeah, so it's by no means universal. I think you echo what the community felt when they saw that, and you're all trials, experts.
Yeah, but so I would ask though, maybe there's no answer. I really, not being in the eye field a lot, I don't understand the y-axis on what's here. I have no idea what quantity of change and from baseline has any real meaning. And so when we say that Oaks succeeded and Derby didn't, I would have to understand clinically or in some other context better what was the presupposed hypothesized difference, because that's what drove the sample size, which is what's driving the p-values here.
Yeah. Yep.
I don't know how to turn that mean change from baseline into anything meaningful for a patient.
Well, I mean, you clearly understand what they did, which was they took the area of the lesions, they looked at how fast they grew, and they're sort of either Pauci or multifocal lesions, generally circular in area, in shape, and they grow. They just keep expanding outward. And so, and you're right, it looks different and it's significantly different, and 21% sounds pretty good. 10% doesn't sound so good. And so, and their sample size was driven by their preliminary data. Right. And I don't know that I mean, what you're driving at is, even if you believe, let's say you believe Oaks and that it works and so forth and so on, you still have to say, okay, so it works. 20%, is that good? Is that bad? Does it mean anything? Is it marginal? And I don't know that people have a good feeling for that. If it has shown a difference in visual acuity, in Derby, for example, we would have, because there you have a higher chance of showing a difference in visual acuity, but you have the less effective results. You would have said, okay, yeah, good idea. Let's use it. But it showed no difference in acuity and no difference in the other functional outcomes. Ian, it looks like you're finally unmuted.
Yeah, sorry about that. Can you hear me?
Yep.
Okay, great. The other thing I was thinking about is, I mean, this is not even within ophthalmology, right, Doug? This is not the first time that drugs have been approved based on surrogate markers of something. I mean, this happens in glaucoma, has happened quite a lot with intraocular pressure. And a lot of the same conversations have been had. So to me, if it was, I mean, the 1 year versus 2 year spot is a tricky one, and I'm not sure where to land on that. But I mean, if we are saying that there is a benefit for a change in this outcome, and we've pre-specified that is an important outcome, you then have to say, I mean, this is a drug that should be approved based on the fact, and something I put in the chat, there's nothing else, at least at the time you're saying, there's nothing else on the market. I mean, it gets into conversations around you approve the drug and do the post-marketing and learn later, the long-term follow-up and things like that. So I mean, it's a tough one. So I would, if I would agree with raising my hand earlier along with the other students who did that at the time, knowing what they knew, and there was nothing else on the market, it probably made sense to approve the drug.
Okay, so can we go to the side effects? Because this is the other controversial aspect of this. And, you know, we picked this article in part because of the Alzheimer's discussion we'd had before. The one that gave other people pause was look at the 24-month cumulative rate of, well, it says exudative AMD, but that's neovascular AMD. That's the stuff where your vision drops right away. And, you know, that gave people pause about the safety of this drug, particularly given the lack of benefit in visual acuity. And they said, well, yeah, you save some retina, but at a cost of giving people neovascular. Having said that, there's always two ways to look at 11% and say, wow, it's a lot higher than placebo, or 11% means 89% didn't get it. And the third way is to say, yeah, it's about 5% per year. So, you know, it became an interesting discussion, and it became an interesting discussion about who was going to use it, particularly in the retina community, for this drug. And there is another drug that's been approved, and I think it was approved, you know, I'd have to look at the articles again, but on the same study design. I mean, and this is where I think Shariza was sort of hinting at this, or one of you was hinting at this, is that, you know, we have established these outcomes, and if you meet them, we will approve your drug. And this is now how we will evaluate them.
But isn't that the ask of the FDA? They're not in the business of regulating medicine, right? So
right.
Effective is effective. Whether it's clinically meaningful is a completely different question, and they're not tasked with that, right? So I think we may get hung up on that too as trialists, because we do want to do meaningful work. But it is important to remember that.
Shariza, can I ask you something for someone who was at the FDA? To what extent does the fact that this is an injection into the eye, this is not just a pill, right? To what extent does that consideration weigh into, here's the minimal efficacy of this drug, but it meets, I mean, it arguably meets the threshold for this interim surrogate outcome. It has harms, it's an injection. So to what extent does the conversation about the fact that it's an injection feed into the conversation?
The mode of delivery is not as relevant as you might think, in terms of, because again, that would be a patient-centered type of outcome, right? So the types of side effects we might expect to see from an injection are different considerations, but they would be compared against other drugs that are administered similarly, right? And so if that safety consideration is a little bit, the harms rather, are a little bit too much when compared to similar drugs, that would be a concern. But in terms of how that might weigh into the overall decision, I think it's taken just as any other safety issue and any other efficacy issue, although the mode of delivery doesn't really factor in too much with efficacy, except you know, for the fact that it's going directly to your blood.
And I would argue that you know, given the fact that now intravitreal injections are widely used and widely accepted, you know, we sort of used to think of them as, oh my God, you're sticking a needle in the eye. But now it's like, yeah, big deal. And you do have to look at the side effects relative to sham. And for the most part, the only difference that you see was that transient issue related to inflammation, and then the new onset exudative AMD. I want to point out one other thing, and this is just a pet peeve of mine, is studies that report the cumulative number of treatment related adverse events and lump everything together. I want you to look at the sham group. 83% of people having a needle, having the hub of a, without actually injecting into the eye, placed against the eye, had a treatment related adverse event. I mean, it's just it's the way we do things, but I think it's a not meaningful number. What I think is meaningful are the very specific ones like exudative AMD and the inflammation and then the endophthalmitis ones. So, but it's an interesting conversation about what to do. Anybody else have any comments they want to make about these articles?
I do want to go back to your question, Doug, if
Yeah.
the students don't have anything to say. Your question about what they would do in terms of whether or not they were reviewers, it's a pretty nuanced question, right? Because
Yeah.
these reviewers are teams of different specialties. So perhaps there are other data outside of the scope of the clinical trial that could have also influenced the direction of this particular approval. And unfortunately, we don't have access to all of that.
Yeah, I agree. Well, and I think one of the things that's important was there's nothing else. And I think Ian in his comment sort of led that. But I think, if there had been other highly effective, if there had been other treatments that were effective, they might have told them to go back and do another one. But given nothing else and given the sum total of the data, I think they were very much influenced by the absence of alternatives and the suggestion that this was, regardless of your estimate of how effective it is, it appears to be effective. And you made a comment about the FDA's job is to just determine if it's safe and effective, not whether or not it should be used. The UK, of course, has NICE, which probably doesn't do its job as well as it should. At least listening to my colleagues in the UK's complaints about it, but they very much look at whether or not the NHS is going to pay for something based upon cost effectiveness considerations.
Absolutely. Lijuan has her hand raised.
Yep.
Yeah, I kind of have a question. I wonder what people think. So I look up on the label and it's a proof saying something about recommended dose for every 25 to 60 days. And like just looking at the curves, there's pretty much no difference between the monthly and every other month. And it's almost slightly, like a few of them, like the every other month looks a little bit better, maybe I'm wrong, but pretty much no difference in terms of efficacy. And then the AEs for that, like the new onset AMD, slightly lower in the every other month. And would you say, I mean, I guess they didn't factor to compare the monthly versus every other month, but like the approval or like indication, would you recommend using monthly at all?
That's an interesting question. I mean, I'm sure that they didn't formally say they were going to, if you look at what they said, they were going to compare the two treatments, right? Not to each other, but compare each treatment, each regimen to sham. And the reason is that if you said you're going to compare those two treatments, you lose power, you inflate your sample size, you do a lot of things that they didn't want to do because they wanted to get the drug approved. The label sounds very much like the people who assisted them with producing the label said, look, it seems to work monthly. It seems to work every other month. We have that. So we are not going to tell you which way to use it. And we're just going to give you a range of that encompasses what was considered monthly in the trial and what was considered every other month in the trial. I mean, Shariza will probably have a comment here because she'll be more familiar with this than am I, but it looks like that's what they did. They just said, both seem to work, or we think they both work about the same. And I actually don't know offhand what people are doing out in the real world, how often they're using it. My guess is, given that after monthly approval of ranibizumab and bevacizumab, they immediately went to treat and extend programs where they tried to increase the interval in order to reduce what they call the treatment burden that a lot of people will be doing every other month, but that's strictly a guess. Shariza, any comments about the labeling?
No, I mean, there's a whole field of research into terms of what's actually put on the label. I'm not entirely sure what the conversation was around this per se, but it's a good observation to make. Don't know.
All right. So you want to go back to your last few points? We have 4 minutes. Maybe we can discuss all the rest of them in 4 minutes. Discussion. Yeah, I mean, we sort of hit these, pretty much all of them on, as we've gone through. So I think that's good. Any last words from our presenters?
No, no from me.
Yeah, I think the previous discussion have include all of the things we want to raise here. So thanks everyone for listening to our presentation.
Thanks all.
Yeah,
All
thank
right.
you. Good
Great
job.
job, all.